All right, guys. Unit 4 is over the cell cycle and cell division. So first we have to ask the question, why do cells divide? And this is on the test. This is to prevent cells from becoming too large. It is when an organism is growing, and it's when you need to replace damaged cells or tissues. So like if you get a cut or a burn, those cells need to be replaced, and so cells will divide to do that. So quickly, we're going to do some differences between prokaryotes and eukaryotes. Now remember, when we're talking about prokaryotes, we're talking about simple cells like bacteria and eukarya. So because they're simple, they have just, their DNA is just one single circular chromosome that's hanging out in the cytoplasm. Remember, they don't have a nucleus. And we have a picture of it below where they've got a circular chromosome. That chromosome replicates because each cell needs a copy of DNA. And then the cell splits in two, and you have two identical daughter cells. Now, when it says identical, that means the DNA is the same and everything is basically the same. This process is called binary fission, and you'll need to remember that for the test. Now, for the rest of this unit, we're really going to be talking about eukaryotes, and that's things like plants, animals, fungus, humans. So the DNA in eukaryotes is often called chromatin, but more often than that, we're talking about when chromatin is packaged tightly and it condenses into these thick structures called chromosomes. And so we mostly refer to DNA in eukaryotes, or often, as chromosomes. These chromosomes are found in the nucleus, and when we talk about the division of the nucleus, that's called mitosis. And below, I have pictures of the stages of mitosis: prophase, metaphase, anaphase, telophase, and cytokinesis. You will usually hear me refer to these as PMAT, P-M-A-T, PMAT. So quickly, we're going to do an overview of the cell cycle. Again, this is in eukaryotes. And it is broken into three stages. We have interphase, and that is when the cell is not dividing. It's the longest portion of the cell cycle. It is the period of growth and DNA replication. Remember, we need to copy the DNA before a cell divides because each cell needs an exact copy of all the DNA. Then when the cell is going to divide, there's mitosis, which is division of the nucleus and of the chromosomes, and then cytokinesis. Cyto means cell, so that's division of the cytoplasm, or the last step in cell division. So here's a picture of the cell cycle. We'll be coming back to this quite a bit. If we look in the middle, that purple area with the white arrow that says interphase, see how it's the longest phase of the cell cycle? Interphase is when the cell is not dividing. We have G1, which is when G stands for growth. That's when the cell is growing. It's doing its normal job, and it is replicating the organelles, and it's getting ready for DNA replication. That happens in S phase, where S stands for synthesis, and we're synthesizing or making something, and that's what we're making is DNA. And then there's G2, or growth 2. That's when the cell is getting ready for division. So interphase, I've just kind of said all this, but you'll need to remember it. G1, you're making new organelles, getting ready for DNA replication. S is DNA replication, and G2 is creation of the parts needed for mitosis. So now we're going to get into mitosis. That's PMAT, prophase, metaphase, anaphase, telophase, followed by cytokinesis.